Charles Darwin, in a famous correspondence with Joseph Hooker, referred to the abominable mystery of angiosperm radiation during the Cretaceous. To him, the appearance and explosive evolutionary diversification of flowering plants, documented by the fossils being collected in America, looked like it was happening way too fast. To his eyes, this seemed like a potentially serious challenge to his model of evolutionary change by natural selection. Certainly, natural selection was powerful enough to account for the range and magnitude of evolution that we saw during this time, but there would still be limits to the speed by which such dramatic change could be effected. Darwin's abominable mystery has subsequently softened to the point of no longer being so mysterious, thanks largely to better estimates for the age of the Earth, which in Darwin's time was much smaller than the 4.6 billion year figure that we use today. Knowing now that the period in question actually stretched out over tens of millions of years makes all the difference in the world as far as Darwin's mystery is concerned. But when it comes to flowering plants, there remains a different mystery, and that is, what happened to this lineage of plants for the 180 million year period from when it split away from the proto-gymnosperm lineage to the first appearance of angiosperm fossils in the Jurassic? To this day, we don't know for sure. There are some candidate plant fossils that have been proposed as sister taxa to the angiosperms, but none of these models is completely accepted. Most of the candidates seem to bear strong similarities to one or more of the gymnosperm taxa, and interpreting them as relatives of the cycads, as is the case with the Benetetiales, or relatives of the conifers, as is the case with the chordites, this excludes them from being part of the proto-angiosperm lineage. Reconstructing angiosperm history is largely an exercise in inference, combining physical evidence from modern plants, from known fossils, and also using a large dose of paleoecology. We can say with certainty that flowering plants have had enormous success from the late Jurassic onwards, with pulses of radiation, that is, rapid evolutionary diversification, starting in the Middle Cretaceous and continuing through the Paleogene. Over this period, global ecology was continually in flux, with a major exclamation point occurring at the end of the Cretaceous when the Chicxulub bolide struck and caused the final extinction of non-bird dinosaurs, as well as many other species and genus-level extinctions across many taxa. Over this period, again from the early to middle Cretaceous of the Mesozoic through the Paleogene of the Cenozoic, the Earth's terrestrial flora transitioned from one in which there were effectively no flowering plants, including grasses, palms, and fruit trees, that is, not just what you might think of as plants with flowers, to one in which flowering plants were not only present, but dominating the landscape, and though not identical to the array of plant species we see today, their fossils were at that point familiar and nicely recognizable. What was the cause for this rapid radiation? Now remember that the increase in biodiversity in any given taxon requires that the rate of speciation, cladogenesis, we talked at length about this in the first unit, be greater than the rate of extinction. Based on what we learned in Unit 1, speciation rate would increase with greater opportunity for allopatry to result in isolated gene pools. Also, if there were a greater diversity of ecological niches into which new forms could specialize and adapt, we could expect this to result in greater speciation rates as well. And thinking more creatively now, maybe there could be some factor making it easier to evolve reproductive isolation. This would also speed up the speciation process. All three of these factors may have contributed to the rapid diversification of angiosperms. First is the idea that early flowering plants may have had greater opportunities to evolve via allopatric speciation, thanks to a highly fragmented population structure. Think about those fruit flies having a speciation storm on the Hawaiian Islands. For flowering plants, this idea is supported by the dark and disturbed model of Taylor-Field et al. Their argument is based on the observation that basal angiosperms, in other words, the ones having the most primitive traits, They're found primarily in semi-shaded habitats like those occurring in forest breaks. Field et al. Start with the premise that the earliest angiosperms were, like their modern relatives, specialists on shady, disturbed habitats like this. Now, in order to connect this with the turbocharged allopatric speciation model, we have to account for small forest break habitats being particularly abundant during the Mesozoic. You know, the era in which there were gigantic dinosaurs? Visualize yourself as a Cretaceous dinosaur. You may be as big as the trees, but moving through the forest is no fun and may be kind of dangerous, because there are T-Rexes around. And so you stick to trails and clearings where the trees have been trampled. This is basically the same behavior we see nowadays from deer in willow thickets or from meadow mice in tall grass. Tiny new forest clearings would occur as a result of dinosaur activity. Every time a T-Rex makes a big mess taking down a hadrosaur in the middle of a forest, trees are going to fall. And such tiny clearings might persist and grow in size for as long as the dinosaurs continue to use them. Left unused, however, these clearings would return to being forested by new tree growth. And this would be the dark and disturbed habitat for which the early angiosperms were specialists. These local patches of semi-shaded forests in different stages of disturbance would have created a geographically discontinuous meta-array of suitable habitats for the early angiosperms, and this would support rapid speciation. So there's model 1. Second, Conrad LaBandera of the Smithsonian notes that the bursts of plant diversification are coincident with bursts of diversification of herbivorous and polliniferous insects. The logic here is that plants and their insect herbivores and pollinators were tied together by coevolution and drove each other's speciation at rapid rates. For example, as the insect herbivores evolved to specialize on eating and detoxifying specific plants, the plants being targeted by the insects co-evolved in response to their herbivores by increasing toxin production or via the evolution of some other kind of defense. Basically, plants and insects were creating new specialized niches as they co-evolved and co-speciated over time. And don't forget about the pollinators. The flowers on flowering plants are not just the megasporophylls and microsporophylls. They're integrated organs that attract and reward animal pollinators. In order to make pollination work best, you want to get into an association with a single guild of pollinators that will visit only flowers of your species. Tighter and tighter associations between pollinators and plants would have driven them to evolve along parallel trajectories. With plants and insects driving each other's evolutions, this increases niche diversity, and it also speeds the rate of evolutionary change between geographically isolated populations. So third, there may actually have been a way to make it easier for isolated populations of flowering plants to evolve reproductive barriers. We talked in Unit 1 about how whole genome duplications were significant in the histories of certain taxa, vertebrates being one and flowering plants being the other. David Tank et al. Show that many of the events of whole genome duplication occurring within flowering plants often shortly precede a burst of diversification for the lineage that underwent the polyploidization. How are these two things, polyploidization and speciation, related? Well, it all has to do with that period of rapid genome evolution taking place in the aftermath of a duplication event. Let's say you've just doubled all of your chromosomes and you're okay with this, but there's still a lot of excess in your genome. And over the next period of your evolution, you can expect there to be a lot of pruning and genomic reorganization. If there are two isolated populations of a recently genome doubled species, the pattern of pruning and reorganization would be different between those two isolates. And this would make it much more likely for the two genomes to rapidly reach the point of being incompatible. In effect, this amounts to speeding up the evolution of reproductive isolation. Now, these are not arguments taken up by most textbooks. My sources here are quite recent, and the conclusions they point to are still somewhat speculative. Their importance to you right now is to provide an opportunity to integrate ideas in the way of a scientist, connecting a real natural phenomenon, here the rapid and bursty evolutionary diversification of flowering plants, with the real mechanisms we discussed in the first unit of this class. The abominable mystery that once concerned Darwin has now become a valuable opportunity to reinforce previous lessons and to integrate new methods and new findings to address old questions.